Hi, Gen Chem 1. This is our experiment for Beer's Law pre-lab video. So we talked a little bit in class about some concepts that are relevant for this, and you need to read some of the textbooks so you're prepared for this experiment. That is section 8.2. Um, it is also maybe helpful to read a little bit of chapter 3 because that explains a little bit about how light works. But we did talk about that during class too. The concept that the color we see is called transmitted light, and the colors that we don't see are absorbed by the object. So my example from class was in a leaf that looks green, the chlorophyll is absorbing the red and blue regions of the spectrum, and the rest that we see is green, right? And so the procedure for this week uses the same information to present how um, how light behaves. And the most important part of that is understanding these words, right? So I told you guys that when we are measuring absorption on the y-axis and wavelength on the x-axis, this is called an absorbance spectrum. You are going to measure one of them for a dye in lab, and you have to use that to find where maximum absorbance happens. That's called lambda max. Lambda stands for the wavelength. And so what this symbol means is wherever we see the highest absorbance peak, in this case in the blue region for chlorophyll, almost into the purple, that is where Beer's law is the most linear, which means the experiment is easier to replicate there. If we measured a wavelength anywhere else, it's not going to be very consistent, which makes it difficult to draw any conclusions about concentration. So lambda max is where we mark the highest absorbance on the absorbance spectrum, okay? That needs to get written down in your notebook because it's going to be used to make the next kind of curve, which is called a standardization curve. Okay, curve and graph are synonymous for us. So a standardization curve is when you take the concentrations of known samples and that's gonna be your x-axis, and the absorbance is gonna be your y-axis. And we looked at an example from our chapter 3 notes, and it's a completely linear relationship where, in theory, the y-intercept should be 0. And so it boils down to this equation I called Beer's law. Okay, now this is all explained again in the introduction of the lab experiment, so it's kind of worth checking that out to make sure you understand what you're measuring. Each group, each pair of people is going to measure 2 graphs. One of them is the absorbance spectrum for the dye you're testing, and the second one is going to be the standardization curve for the dye you're testing. Then you're going to share those graphs by sending them to your partner as well as 2 other people. So by the end of the day, you're going to walk away with 4 total graphs, 2 absorbance curves, 2 standardization curves, but you're only responsible for measuring 1 of each. So pause the video here and take a second to answer the first pre-lab question. So in order to create the second graph, you have to have calculated the concentration of the known solutions that you're going to measure. This information gets entered into the little computer that you're going to use. It's a little tablet called a LabQuest. But the equation from chapter 8.2, which starts on page 343 of the textbook, is C1V1 equals C2V2. So I'm going to do an example of that, just so you have it. But what you'll have to do for our specific experiment is you need to look through the procedure and see what is our stock concentration. It's listed in the procedure. We need to be able to do some unit conversions from the stock units into something more useful to us, which is going to be micrograms per milliliter. This is part of why we practice dimensional analysis and metric unit conversions in chapter 1. And then the initial volume is what we're going to solve for. The final volume is listed in the procedure, and I'll show you where that is in a moment. And then the final concentration is what we're solving for. Okay, so for example, I start with a stock concentration of 35 grams in 3 liters. My first step in this experiment would be to convert that, the grams into micrograms and the liters into milliliters, and then put those two together to get a concentration in the correct units. Okay, so given I'm going to do the 35 grams first, I need to know that there is 1,000,000 micrograms in 1 gram. Okay. And then I'm going to put this in scientific notation just to give myself some space, but so that's a lot of micrograms because 35 grams is a lot. And then I'll take the volume in liters, so 3 liters, and convert it into milliliters. There's 1,000 milli for every 1 base unit. So that's going to be 3,000 milliliters. Okay. I'm going to change color my pen so I can find my answer easily. But we're going to go the micrograms on the top and the milliliters on the bottom. And that will get us the correct units for our, right? So my answer to that part of the calculation, it says 11666.7. I'm going to round because I only have 2 significant digits in my original information. So I'm going to go ahead and round it and write it in scientific notation. My unit is microgram per milliliter. So that is one part of what you need to do for this experiment. So calculate on your lab notebook pages, write down your dimensional analysis to calculate the micrograms per milliliter of that stock solution. Okay, so the last step is to use this information about how much stock is being added and how much water you're going to add to calculate the concentration C2 of each sample you're actually going to measure. So remember that V1 is the amount of stock. So the first 10 milliliter dilution here is not, it's not really a dilution, so there's nothing to calculate there. You already calculated the micrograms per milliliter for that in the last questions. But the first dilution you're actually doing is 9 milliliters of stock with 1 milliliter of water. That's going to use C1V1 equals C2V2, where C1 is the stock in microgram per milliliter. So the answer to the last calculation you did. V1 would be like 9 milliliters. C2 is what you're solving for. And V2 is the total volume of stock and water together. So 10 milliliters, okay? You don't have to convert to liters or anything like that. Everything can just be in the units, microgram per milliliter for concentrations and milliliters for volume. Once you have the answers for all of the dilutions, write them down in your notebook and enter them in the quizzes app here to see if you have it correct. And then you're ready for lab.